Yeah, hello everyone. Welcome back to the bioengineering course. What we will today look at is actually plasmid design. So we have learned a lot of things, right? We have learned from DNA, from translation, from transcription. We've learned how to generate primers. This will be very important today, and especially for the problem session. And today we are creating our first little program completely ourselves. So I think this will be a nice milestone. Yeah? So what is this actually about? So let's get started why we do that. Yeah? So the purpose of this session is kind of to write with you together a Hello World program in DNA. So we have to first think, of course, how do we write that, right? And there is different versions. Like the perhaps most direct version would be we just write the string of DNA together, right? Like, I mean, this could be like essentially you just tell me, please put this sequence, synthesize it, and please put it into the bacterium, and this is my Hello World program. Yeah? I mean, of course, we are going not to do that. This is first, I mean, it's out of reach for even very simple things, as we will see. Even our Hello World will be super complex. Huh? And second, of course, it's completely error prone. Huh? So the second thing that probably most researchers are doing is actually to use some other programs like either specialized programs or even Excel or something, yeah, just to design by copying around strings essentially, or clicking and dragging and dropping around things. So let's call this something with a GUI. Yeah? You're creating your plasmid. So in the end, you have a plasmid. By, let's say, you select this part here, then you copy it here, and so on, and so you process, and in the end, you have what you want. And this is hopefully what you have. And this is the thing, the string, the DNA string that you're going to synthesize or to use, and you put this in the into the bacteria. We are also not going to use that here, or so it's perhaps the most common way to do that. We are actually advocating something different in this lecture, and this is code. So we will use code to create code. I mean, this is a very old concept in computer science. Like it's computer science is doing this from the very beginning, right? Like when people are not writing in machine code, people are normally not writing in assembler code. You write compilers, and we are perhaps not doing a fully fledged compiler here, and it is out of the scheme for this thing here. But we are still kind of generating with Python code. We are generating the DNA code. This is the concept here. Yeah? And so why would anybody do that? Is there a question or sound? Everything fine. Okay, good. Sorry for the interruption. Then, like, why would anybody do that? Yeah, right. I mean, this seems out of reach. This seems quite a nice idea, actually. I mean, it's just what we're used to, but dragging and dropping things around. And why would anybody try to code that? Huh? And we think there's actually there's many advantages to this, but in particular we want to highlight two advantages to this. Huh? So the first is actually that this is kind of helping to automatize it. So automation. Yeah. Your code that you generate, let's say in Python, to generate this, it might be not totally general. So it might be not a compiler that compiles any, I don't know, Python code into DNA. This is out of reach. Yeah? But still, hopefully your code is written in a way that with only a few changes, and if the design is quite close to what you already had, you can still get something. And here you would have to run through all these steps again. So this is the first big thing. Yeah? But there is a second thing here. This is documentation. Like code is documentation. Yeah? If you give me, for example, your nice spreadsheet with here the plasmid inside, yeah, I have to totally trust you. I mean, there's no easy way for me to verify or to redo it perhaps myself, to learn from it perhaps what you did. Yeah? I have to kind of trust you. I mean, you can give me, of course, all these documents, yeah? but still, then imagine I do a mistake from clicking from here to here. I paste not this sequence, but I paste this sequence. I'm actually doing a mistake. This is much more error prone, and this code is really, really a very great tool to kind of precisely describe what you want to be done. Code is marvelous for this. And very related to it, so perhaps even the same point, is verification. So here of course, you can get arbitrarily complex with verification, code verification. What we did in this lecture and what we show here is kind of putting nice checks and assertions at the right places. It's not totally verifying that your DNA will be out of reach to work in a bacteria, but it's of kind of, you think, oh, this should have actually the same length as this, or this should be homologous or complementary to this. So let's just put an assertion here. So your code should be full of such checks. So we strongly advocate this here. So let's do that. Let me say something else. Let me perhaps specify, because this I didn't tell you yet, what will be our hello world. So what's the purpose of a hello world program, right? In Python, it's super simple. I mean, you have your main pie, right? And you just have a print hello world. It's the concept of a hello world program. So what it does is, if you call it, it just says hello world. So it's a super simple thing that you just demonstrate not even all concepts, but just one little concept of a coding language. So what will be this in our case? I mean, we cannot yet make bacteria that write hello world to a screen. That would be not a hello world program if you could. It would be a very nice paper. So what will it be? And we thought hard, and we had the idea of why not using a color? I mean, this is like, you have here a pickle dish, and now I particularly brought this here. You have some spots of E. coli colonies here, and we want, where it worked, we want them to be pink. We want to see kind of a hello pink world kind of. So this will be our hello world. Sounds very easy, but you will see actually it's already involved to do this in code. And I think we see a lot of concepts when we just do this very simple cloning and this simple design. Okay, so now that we are set up and we have the problem, we can start designing. Yeah, by the way, in particular, when we talk about a design, we don't want to include this DNA code into the genome. That would be, again, too complicated. We just want to create a plasmid, right? A circular DNA. That is circular. So, of course, double-stranded circular, right? And that we want to include into like to transform into a bacteria, and then that this bacteria shows up pink. This colony shows up pink. Okay. Very quickly, if you check out our lecture webpage, we have uploaded a background material that is quite important to. I mean, I will explain the basics here, but if you are interested in the details, check this out. It has a quick description about what is a protocol. Kind of, this is an experimental algorithm. So what you have to do in the wet lab to achieve for what you want, for example, to extract an A, to clone, or to stock and grow bacteria. We did a list of the things you will need for this hello world program, and some other things you might need there. And in particular, we have detailed on one particular protocol. This is here, cloning of a plasmid, because this is the big thing we want to do. And what does that mean? Cloning of a plasmid will be actually, we will design such a plasmid, we will multiply it, and then we will insert into the bacteria, and then grow them again, and then they will be showing up pink. And this is actually this protocol. So it's describing how once you have this string of, even here concretely, once you have this plasmid, what you're going to do, and also how to create this plasmid from the parts. Yeah. So yeah, you will find details here. Perhaps one thing I want to say, just a very few words about this, or let's look together at these figures. Yeah. A very big part of this thing is actually Gibson cloning. Yeah. So how we are going to design that is actually we will take a backbone. So we will have a plasmid, yeah, that has kind of the basics on it. What are the basics? This means this includes the origin of replication. This includes perhaps some antibiotic resistance gene, a promoter that expresses this, and so on. It's not super full, but it has a few things just to kind of survive. And this plasmid is typically called a backbone or a vector because it's thought to have afterwards an insert, to carry an insert. So what we will actually do is we will kind of linearize this, and you will do one method to linearize this in the lecture, and you will do another method in the problem session. So linearizing means just creating a linear version out of this. Okay. And then we will design an insert. So this is a second sequence here, painted here. And then you will have to kind of glue together here and here the insert with the backbone. And then you have, in the end, you have the plasmid you want. And how do you linearize this? There is two big concepts to linearize something. So one is to cut open with restriction enzymes. So it's an enzyme that cuts at one or multiple positions that it recognizes in the DNA. Another would be to PCR it, like you saw that in the last lecture, how to design forward and backward primers. You can do the same with a plasmid to linearize it, right? So if this is your plasmid, and if you add, for example, here a forward primer and here a backward primer, then you will have actually this part of the plasmid as a linear version, right? So you can even remove stuff. If you would choose them like here from here, then you can have the complete one linearized, of course. But you will use it actually to cut something out. And then the question is how to do this gluing together, right? Once you have this linearized version of the backbone and you have this single fragment here, like we will have only a single insert. You could have multiple, but we use only a single one. I'll just close this a little bit. Then we will do this. There is different protocols to do this. And here in this background material, there is one described which is called Gibson cloning. And how that works, you find the details here. But the principal idea we see here, it has actually three main ingredients. Of course, like also the nucleotides and so on, but three very important things. And the first one is actually a 5' and exonuclease. So that's taking your linear fragment, which in your case will be the insert and the backbone, and it's chewing back from the 5' end basis. It's keeping this 3' end, it doesn't touch that, and it's chewing this back. So this results in actually sticky ends, so-called sticky ends or overhangs. So 3' overhangs. And in a second step, but this actually the cool thing about this Gibson thing is that this is all in parallel, but we can think of it in a second step. This start to anneal, of course, only if you have designed it in a clever way. That a clever way meaning same principles as you heard last lecture with the primers. Just design a complementary sequence, then it starts to anneal. Typically, you are aiming here for around 20 nucleotides in overlap. But this depends a bit on the protocol and design. And then the DNA polymerase that also is in mix starts to fill up here again. Because why can it start to fill up now? I mean, we know that DNA polymerase works from the 5' to the 3' direction. So, and it needs a template strength. So everything is here. So it can just do its work. It cannot completely do its work. It has to stop here. So there exists still the nix. And this is the last magic that's happening. There is actually a DNA ligase in the mix. That's just simply then healing the mix, and that's your constructor. Yeah, all the details you find in the background material. Okay. Any questions perhaps to this? I mean, I know this is super short and you have to read this. I mean, it's, yeah, give it a read. But if you have immediate questions, let me know. Otherwise, we can proceed to the main part of the lecture. Do we have some questions or is it fine? Okay. Yeah, okay, good. Let's be to interrupt when you want. Okay, we will use some old libraries and new libraries to do that, right? I think you already know BioPython from last time. I'm going to use this heavily, in particular for restriction enzyme simulations. DNA plot lib, you already know that from the from the gates. And we will use the new ones, DNA features view. This is a very nice thing to look at annotated. It's a kind of a sequence viewer for annotated DNA. Very, very nice. And PyDNA, we will use this to simulate Gibson cloning. So this will be very nice because this is kind of the verification that I wrote here. Like you will design the backbone, you will design the insert, and then you can simulate with PyDNA whether this really works in a correct Gibson assembly. That's a pretty nice tool. Yeah, we will do also some digesting to this here. This is much more for you to read. We will also do some logging because our code will be a bit more complicated. So this is a nice habit, of course, to instead of cluttering everything with prints. Okay, so I think this we had already looked at. So what will be our design? What's our Hello World program? And I guess it will be looking very simple, kind of like the circuit diagram is super simple. We have a promoter. In our case, this will be an inducible promoter because it's already on the backbone. That could be a constitutive one, completely fine. It will have an RBS. Detail on this a bit more, what that means. We will have a pink protein, yeah, of course, to show up pink, and we will have a terminator, and that's it. Super simple design. Yeah? That's our Hello World. So the first thing we have to choose is we have to choose a backbone. So that means you have to go to the internet and pick one of your favorite plasmids there. There is a huge, I mean, there's many websites with many libraries. It's an art itself, like how to choose the right backbone, yeah, for your technique. Of course, with choosing the backbone, you have to choose also the right host bacteria, right, the right chassis. But we will stay very simple here, very traditional here. Like we will pick very simple bacteria, and we will pick them that they work actually with a plasmid that is very heavily used. So this is the PUC19 plasmid. So I think we have that in our lab, for example, and most labs have that probably stocked. And if not, you get it very easily from Edgene, for example. So something like this, PUC19. Small p normally means plasmid. Normally, if it's a big P, it's a promoter. Small p, I mean, there's always exceptions, but that means plasmid. You see here in this case, so this is means University of California. 19 is just a number. I think they just kept increasing the number when they kept increasing the plasmid versions. There is also using 18, for example, and this is 19. Of course, this is not standardized. Yeah, I mean, all the plasmids have different nomenclatures. Yeah? Okay, so let's get that. We will concretely get, so annotated DNA comes in very different flavors. We will actually get something that is called a gene bank file. Yeah? This, opposed to a FASTA file, which normally has just the DNA and very simple annotation. This is really annotated. Of course, it's always as annotated as the one who wrote it annotated it. So, but this is a pretty good annotated one. And we will get that from Edgene because let's say we assume we order it from Edgene. So why not get their annotated file to be sure? So let's do that. And I don't walk you through the code here, but this uses the DNA feature viewer to actually look at DNA features. We have just added some code around it because we want some like not DNA features, but coding features. Yeah? For example, to crop some sequences, to zoom in, to zoom out, and all this. And we had to do some fixes, and this is all here on the website. We will perhaps also at some point write a library with such simple functions that you can use, but otherwise at the moment you find it here in the cell. So did the coding for you here. It's a bit lengthy. And here we can use it. So this is how you use it. You just tell it, do you want to plot me as a cycle or as a linear? I mean, in the end, it's a cycle, but how do you want to plot it? And you just say, here you put the sequence, and here's some parameters, and that's it. Huh? And if you do that, and I don't collapse, why is this collapsed? In a particular, that's how I uncollapse it. It prints that. So that's our backbone. Looks, I mean, for a backbone, looks pretty full, right? But it's still quite empty if you look at it. Yeah. So what do we find here? Let's go through it. So here we have the origin of replication. I mean, that must be present, right? Here we find another construct. This is a very simple construct. It has just a promoter and a gene. And this gene, what it's doing, it's called AMPR. So you can look that up, but this is essentially an ampicillin resistance. Yeah? So whatever bacteria this is constitutively expressing, it will be resistant to ampicillin. And this will be very nice to select the bacteria. For example, if we put here ampicillin, right? We are sure to have only the bacteria that carry this plasmid and not the others where it got lost. Okay, so this is what remains, these two things. Yeah? And so what is that? You might perhaps even know that from previous lectures and homework. Because you recognize a luck promoter and some luck set here, which is a bit strange because it has a luck set alpha, but I'm going to explain that. Yeah? So in the end, what you see here is you have a promoter, which is in particular very nicely annotated, I have to say, because they even annotated the cap binding site. You remember it had two binding sites, yeah? The cap binding site, and here it has the operator, yeah, for the repressor. So really beautifully annotated, and this is the promoter. What is a bit strange is this thing here, why it's actually a luck set alpha, and that hints actually that this is a fragment. Yeah? It's not a complete luck set, it's only a fragment of the protein. So why would you do that? I mean, this is very strange. Huh? And I can perhaps draw something here. So this will be a really nice feature, also why we choose this plasmid. It allows you to do something which is called blue-white screens. And that means the following. Here, let's say, without our design, we are just taking the PUC 19 and putting it into bacteria, nothing else. What will happen here is actually that if you choose a certain type of bacteria, in particular a bacteria that has the other part of that luck set, yeah, the omega part, yeah, the behind, this is going to kind of glue together, yeah, and form the kind of complete protein, yeah? And that means you are giving, with this plasmid, once they have this, they can form the luck set, and they will use the luck set that in presence, if you grow them on, of course, let's say LB, your growth medium, plus if you give them XGAL, this is kind of a dye, we will see in a second, yeah, plus the inducer of this promoter here. So we know that this can be induced by IPTG, right? You know that from your program session. Then actually it will synthesize like that, and this will, because of this XGAL here, it will form blue colonies. it will show up very dark blue, a really nice dark full blue. Yeah. So I mean, now you can say, well, why do you need that? I mean, there's another test to select the colonies that don't have a plasmid, right? And that was just, I mean, we just had that. We just put ampicillin. So why would I need double selection, kind of, yeah? And the magic is this thing here. The magic is actually there is a sequence MCS. Yeah? This is a so-called multiple cloning site sequence. And this is a little sequence that the plasmid designers from University of California designed in a way that it's completely full of restriction enzyme sites. It's packed with such sites. We will see them concretely in a second. And they have the special property that they occur only once here and never outside. Yeah? So whatever restriction enzyme you use, you cut it open exactly once here and doesn't touch anything else here. So it's particularly made to be inserted here, your construct later. Yeah? We will linearize it here and we will put our insert and our construct in here. Yeah? Okay, but this is very weird, right? Like, I mean, this is overlapping completely with this fragment here. But that's the point. Yeah? Because actually, if your insert works, you're totally destroying this blue thing. You will have a white colony, right? So in the end, you will have kind of 3 types of colonies after the insertion. You will have the colonies that don't have the plasmid. I mean, they're immediately killed by ampicillin. They don't grow. You will have the colonies where your cloning didn't work. They are blue. And you will have the colonies where it hopefully worked, and they're white. I mean, this was a pre-sequencing thing. Like, it's a very simple test, but it's a super nice test to pick colonies that have the construct. So really cool thing. And in particular, if you look at them, I think on Wikipedia there is a picture. I mean, you can very easily see, this is how it really looks. You can very easily distinguish the white and the blue colonies, and you're sure you have an insert here. Yeah? Well, at least something inserted. Yeah? Yeah? So works really, really nice. Check the time, okay. Okay, so I think we can progress. So I promised you that this MCS sequence here, right, that this is full of restriction sites that are existing somewhere else. And this is essentially, again, this is the cool thing about code. I mean, of course, you could Control F your sequence that you got from Wikipedia and looked at it doesn't appear, but how do you convince the others that it's this, and so on and so on? Here we just code that, and we write a function that just checks if it occurs only once. Okay, these were some other checks, by the way. This is yeah, sorry about this. This was some, we added some other annotation. We will going to need this, but I skipped it for the moment. This will be important later, but we just added the start codon of this LuxSet alpha fragment. We also annotated that for a reason later. Yeah, but here, and now I come to what I promised you, to linearizing the backbone, and we are going to do that. We're looking at this MCS feature, right? It's here. Here it also tells us what it is actually if you look at the multiple cloning sites. And here, this is now the function I promised you, the get and temp that only cut in the feature of MCS. And you just put the feature and it tells you it doesn't cut out. It cuts only here. So this is this. We can run that, right? And here it is. Here's our answer. That's the list of things that cut only inside. Yeah, and that's that's a huge list. I mean, it's by design. Like the people who did the construct did this on purpose for us. So super nice. So we can pick essentially any one of them to linearize, right? Whatever you want, you can pick it. There is some who are very difficult to do in the lab. There is some who is super easy to do. So you should not pick perhaps the first in the list. So how are we gonna pick it? I mean, let's first watch that. So, for example, here we're using BioPython to show where actually in this MCS sequence things cut. And we just tried on some things that we selected that are known. So this list is ones that are quite nice behaving. I mean, echo R1 is perhaps the most common one and nicely behaving and very easy and probably present in every lab. Yeah? BAM H1 is also a very nicely behaving one. We will pick it here for a small reason over EQBR1 that I'll show you in a second, but otherwise it's completely fine. Yeah? And actually, we can see this small reason already here. Like if you see here, you can imagine here is your promoter. Here is the coding sequence we are going to insert or uninsert. So the only thing I chose BAM H1 is like it's a little bit closer to the promoter. So whatever we're going to insert here, and this will be the ribosome binding site, we want it to be a little bit closer to the promoter, but it's totally fine to do that here also. Okay. Yeah. Here we use an annotation of these two cutting sites just to look at it. Here you see that very nicely, right? Here we have the promoter, here we have the operator, and we can choose between this and this. Both are fine. And here's our annotated start codon that we will second use that. Yeah, now we use this very nice feature of biorestriction to visualize a cut. Because at the moment we just know it cuts at this point. We don't know anything about how the cut looks like, right? I mean, cuts are often not blunt. Blunt meaning like here we have the DNA, right? Here the 5 to 3, 5 to 3. Cuts are often not that they are like this. Yeah? These are blunt cuts. They exist, but typically a bit more difficult to treat. Cuts are often in the way that they look like this, right? So there is some overhang, and we have to take that into account, this overhang. And so let's do this with a little bit of coding. We wrote some code where you can say echo R1 and it tells you in your concrete sequence how this will look like. Very nice. And for BAM H1, same. Like it looks, they have the same overhang. They have the same 5' overhang. Behaves really the same. So we are really free to choose. Let's just choose this BAM H1 here. Yeah? By the way, is this a problem? I mean, I told you we should linearize, and then we have to make this homology for the Gibson cloning here and here, right, with our insert. The homology should be 20. Right? Is this a problem that it's not blunt here? Any ideas? Also online viewers? If this is a problem that we have an overhang here, that we are not clean cutting here. Yeah? Would it have been better that we use a blunt cut, or just cut it and then we have a very nicely linearized DNA, or don't we? Yeah, no, the snapping back is not so much. It depends on the temperature, of course, and you can control that. So remember there was a 5' exonuclease in our mix, in our Gibson mix. And what is it? A 5' exonuclease doing? Like it's yum yum yum, or it's eating from here, from the 5' end. That means it starts eating to the inside. Yeah? So even if we cut like here blunt, a few seconds later we would we would have been eaten inside anyway. Yeah? So there is not a big deal. And so of course you could have make blunt cuts. Yeah, no problem with that. But the only thing is like the enzymes are much more. Like the efficiency is a bit less, they're a bit more tricky to treat and so on. And so this is the reason why not to do this here. Yeah. But you could, of course. Yeah. Okay, so we kind of did the cleaning here. So this is conceptually after the 5' exonuclease has eaten away the overhang. Yeah? Of course, imagine this. The 5' exonuclease doesn't stop here at the blunt end. Of course, it continues eating inside. Yeah? But like computationally, it's nice to have a blunt end here because then you can easily take the complement from this as this and so on. Yeah? But it's like after making it blunt kind of. Yeah, for the Gibson cloning, what we need is a homology length of around 20. Yeah? This is the protocol who tells that to us. In fact, it depends on the number of fragments, depends on the length of the fragment and so on. But with 20, you almost always quite well for a few fragments. And we have one fragment that we want to insert, so completely fine. And of course, now that you have this is the sequence upstream and downstream of the cut, it's very easy to get a 20 homology from this, of course. Just a few lines of code again. And again, like this is again, I cannot point this out. I think this is perhaps the thing about this lecture. Write it as code, right? I mean, of course, I could have just copied that. With a mouse, I can just copy 20 of this, and it's done. It's seemingly easy to do that, and perhaps even quicker first. But I mean, who tells me that my mouse shouldn't have gone until here or here? Yeah, or who tells me that I don't know? There was an email in between. I don't know what. Like, or imagine like you want to run it afterwards, not with BM, so BAM H1, but you with EcoR1. You have to select it again with the mouse. Like I think write everything with code. And we did this here. Okay, let me just give me a second. And check the time. Okay, no, we're good in time. All right. So now that we have what did we do? We have our backbone. We have cut it open, and it's now linearized, right? With this part being, let's say, here, and this part here being here. Now we need our insert. We need the thing that we have to stick together to this ends here. And how does our insert look like, right? Our insert looks like a promoter, a ribosome binding site, pink gene, like that will be, we'll see this in a second, what that will be, and a terminator. And then, of course, we have to end here the arms, right? So we have to add the same, or yeah, probably we have to add the arms. Sure, that they can finally anneal to the constructor. So what are we choosing? Wait, sorry. I made a mistake here. Let me correct it. We don't, of course, sorry about that. We don't need the promoter because that's already waiting for us on the constructor. Like, yeah, don't know. Yeah, I mixed up here the directions, but it's waiting here outside for us. Yeah. Okay. So what are we going to choose? How do we choose the ribosome binding site? Right? What's a good idea for that? And this is it's not that easy. It's not that easy. So first we have to think about what does a ribosome binding site contain. Yeah. And if you give it a search, you find something that is called a Shine-Dalgarno sequence. Yeah? And this is something that has been found by these two people, yeah, that have found out that actually in E. coli and other organisms, there is a sequence that, of course, has little variations from time to time, but the consensus sequence that you if you average and look at the statistics of the sequences, appears the most often, and this is this sequence in RNA, of course, yeah, because it's for translation. And if you map it to DNA, it would look like this. It depends a bit on the organism. So for E. coli, it's this. For humans, it would be a bit different and so on. And so that tells us we have to have this consensus sequence, right? So we are now zooming into this here. We have here this Shine-Dalgarno sequence. Then we have to here have a start codon. like I'm writing this now in DNA, so it would be AUG in RNA and ATG in DNA, right? And so first, it's typically not a very good idea to take exactly this sequence here because this is super strong. Yeah. So people have fortunately did some experiments, and actually some very nice paper exists also about some computational aspects about how to do this. Yeah? And we pick actually one of the sequences. Yeah. That is very well known in synthetic biology. I think it has been used by Ilowitz. Yeah? And this is this famous BBA B0034. Yeah, so that is in many designs you will find that, and this is a quite strong but not very strong Schein-Delago variant that we will use. So it's this one concretely. If you look at it with a bit eyes closed, you will see the consensus sequence here. Yeah? It's roughly very close to the consensus sequence, but also not identical. Yeah? And then we have to add something. Yeah, we have to add on the left side. We have to add what is called a standby site. Yeah. And on the right, we have to add something which is called a spacer. Right? And again, there have been many experiments by really good researchers about what's the right spacer. And for the spacer, it came out that it's not so important the sequence. I mean, of course, you should not put like perhaps 5 Cs or something, yeah, or something very sleepy, 5 As. Yeah? But it's not very important what sequence you put here. Much more important is the length of the sequence. And the optimum has been found to be 5. Yeah? You can vary it with a few, let's say 4 to 8 is quite completely fine, but then it starts to degrade a bit. Huh? But 5 is the optimum, I think. The paper is linked here. And about this here it's quite important that this becomes accessible to the part of the ribosome. Yeah? So here the sequence is important, much more the structure that this is forming is important. Yeah? It's kind of isolating the rest that's coming here, the untranslated region from this to be accessible, kind of is the intuition. And again, there is a very nice paper that is also here linked here by Salis, Mirsky, and Voiter. It's linked here that you find really a detailed discussion of all these parts and how to construct it computationally in a nice way. Yeah. Yeah. But here we made it simple. We have just picked this one, and that comes already with these parts. Yeah? So we are fine. Yeah. And if you look, for example, at the spacer here, there's 1, 2, 3, 4, 5, 6. Not optimum, but pretty close to optimum. Yeah? So we are going to annotate that with BioPython. Sorry. And adding it to our insert. Yeah? Like we are creating an RBS sequence that has this annotation and the sequence insert. Yeah? Okay, now that we did this, we are going to do this. This is actually pretty easy also. The not so easy part was actually, funnily, to find the sequence. I had very quickly what I wanted to use. In this case, it was the M-cherry protein you need to express, but it was very tricky to find the sequence online. I don't know why. Yeah? And yeah, in the end, of course we did it, and it was available. What Thomas has shown you last time. So you use the same control with this entrées. I think it's called Uniprotor, like this library, and you can get that. You have just to find this magic number here. Here it's the M-cherry gene. Because yeah, what was very easy to find is I found very easily the protein sequence, but I wanted to have the coding sequence. But yeah, we found it, and here it is. We are downloading that, and we are also annotating that right away. Because like whenever you have something, whenever you bring something immediately annotated, then you have that nicely in your viewer, right? And yeah, we are just checking. Like that would be a small assertion. I didn't put an assert here, but that would be a small assertion you can very nicely put here. We are translating it, and this should always start with an M. Yeah? Like not always, but if it's not, you should have a good reason. By the way, this M added something here. This stands actually for met in long or methionine. And you can check out that this is post-translationary. This is almost always or very frequently removed. So in the final protein, it's not there anymore. But it depends a bit on the next few amino acids, if it's being removed or not. Like there are some heuristics. I don't know if in this protein it's being removed. I think yes, but didn't check that. But you should always find this M in the beginning. And another thing we should always find, unfortunately, yes, it does that. We should always find a star. That means there's stop codon in the end. So everything fine here. So we did that. Next is the terminator. So for the terminator, we also look in the big library of the internet. And in this case, I found a nice one in iGEM parts, which, by the way, was difficult to access because they are just reworking the library. So some of the link was stored, but okay, here it is. So this is also a very famous terminator. It's a double terminator that is very frequently used. For some reason, didn't find an API for iGEM that was working at this time. So here I really copied, shame on me, but we had to make it work. So here we copied the sequence, and here we annotated, and this is the terminator. And here it is. So now, how do we glue together the sequences? I mean, very simple. We just put pluses, and everything is annotated. So the total is also annotated. So we have an annotated insert. Very nice, right? Here are our homology arms, right? This is just a 20 left and 20 right. This is this here and this one, right? We have to add that also. And here we just look at it. Looks exactly as expected. Everything is fine here. Yeah, and you can even see the homology arms, the little things here on the side. Okay, now the time, yeah, still good. Yeah, yeah. The terminator is to stop transcription. Yeah, yeah, exactly. Yeah. So we actually, I mean, you would not necessarily have to edit, because I mean, you're just wasting resources here. Yeah, because I mean, for us there is no construct which comes right after, so probably anyway at some point transcription will stop. Yeah, but it's just good habit to say. I mean, it's a waste of resources if you keep transcribing from there. Yeah? So we just stop that here. Yeah. Yeah. So translation stops with the stop codon, and transcription stops with kind of a terminator. Yeah? Yeah. Okay, so we have everything together. We have what we think is an insert. We have what we think is a good position to cut. And now comes the magic of code. Yeah, we can just simulate that. Yeah, so let's try that if it works out. I mean, we don't know. So there is this library called PyDNA, which does exactly what we need, which has assembly functions, and we are just saying, here is the backbone. Here is where we want to cut. We are cutting actually this thing. Here is our insert sequence. We call it insert. Our fragments is the linear backbone and the insert. Please assemble it and tell us what comes out of it. Yeah? Really nice. Huh? And it's taking care of sticky ends and whatnot. Like this is a really cool library. And it tells us that there is a final construct. So that worked. And now we are going to look at this final construct. So first we have just printed the promoters. That was just for fun, like to see, oh, are our two promoters still present? Yeah, and they are. Yeah, surprise. Yeah. But much more interesting is perhaps we zoomed into the promoter that's really interesting us. So we zoomed into this construct here, just the little part end of this promoter. Yeah, in particular, its operator side. So the very last base. And here we just took the ATG, so the start codon of the MG array. Because here, this is the most critical region, right? Here we can do the most of the mistakes. Yeah?
There is a question online. What do you recommend? Python-based cloning simulation like this, or software like Benchling or Snapchat?
Okay, so I mean, I have a strong preference, and in the spirit of this lecture here, I would really use Python-based. Yeah? I know that Snapchat and Benchling has that, yeah, and they are doing also a great job, yeah. So I don't want to say bad things about this, but the problem is here the really nice thing is about that everything here is coded, right? So you can just, in the end, I can just run play here, and it tells me if everything is correct. Yeah? To my understanding, what I would do in some other softwares where there exist many, I would, for example, have to take this sequence and do a simulation there. Yeah, and of course I can do that. Yeah, but I could not add some other assertions, for example, armor two promoters here and so on and so on. Yeah, so I would really advocate the code usage here. Yeah? Hope this answers the question. Okay, so this is our construct and where we zoomed in, and everything looks fine. There is a little not nice thing, but that was on purpose for a Hello World example. We were a little bit cheating, right? Because, and this is the reason why I kept that insight from the beginning. We have the start codon of the original sequence that we have now cut in the middle, right? Of course, it's here. It's still here, right? But we have just annotated the starter. So what is probably present here, and this you will find that out in the problem session, yeah? There is probably a Schein-Delagno sequence here. Because, I mean, it was working, right? There was the operator, there was the start codon, so probably there is a ribosome binding site here. And I can give you this hint, there is one. Yeah? So what does that mean? That means actually ribosomes are going to bind here and start translating here. Yeah? And some other ribosomes are going to bind here in our one and start translating here. Yeah? So we're kind of a little bit wasteful here, right? We have now two ribosome binding sites on our mRNA with an even smaller fragment, garbage, yeah? And then the real thing. Yeah? So of course, many will actually go here and do what they want. So our colonies will show up pink. Yeah? But we will also waste a bit resources here. Yeah? Yeah, you will. I don't want to say too much word about it, but the reason why we did it was we wanted to cut with an enzyme in the MCS sequence, and that was not present here. So there is no cutting here. But there is another technique not to cut with enzymes, but to cut with PCR, and you will do a very clean design in the problem session. So you will go exactly from here to the left, and then you don't have this problem. This is the final plasmid. This is if we look at the final construct. You see how simple it is. We just, perhaps, yeah, this is also one thing. You don't have to click anything here. You just say plot sequence final construct. And I mean, look at the, this was the complete simulation of the Gibson, including cutting. So it's really super simple, and completely automatizable. You don't have to copy, cut any sequences with a GUI. And that's our construct. And now you can look at it and look at it if you're happy. Of course, again, you can run tons of verification code here. Yeah? Didn't do this here, for the lecture purposes. But you can check, is the MR still here? I don't know, is the plasmid between this and this size? Do we have additional start codons from here to here or something? Yeah, you can do all these checks automatically here, and not with Control F look in your sequences and I know your next colleague forgets to run these checks. Yeah, this brings me to the end of the lecture. So what we have seen, and I think this is the big takeaway, we have a pure Python notebook that just generates and verifies and simulates a Hello World program in DNA. Yeah? And yeah, hope you liked it. you have all the constructs here. You can go to the lab and try that out. Yep. Thank you.